sincere thanks to Philip Reid, President of the Brown Breeding Society of Zimbabwe, for stimulating this, for pushing this. It started last year in October. This is just an introductory course into what we expect back from the stud breeders to make sure, as Philip mentioned at the beginning, that all the data is up to speed, and we are indeed in the business of stud breeding, and everything is up to speed. So the first thing I just want to touch on, ZHB. What is ZHB? So Zimbabwe Herd Book is the data registry maintaining the herd books on behalf of all the breed societies. So the breed societies all have constitutions, all have bylaws that fit under the overarching constitution and bylaws of the Zimbabwe Herd Book, and they dictate what a stud animal is, what are the requirements for an animal to enter the herd book, what are the requirements for the animal to stay in the herd book, to be moved, to be upgraded, whatever. And basically we maintain the herd book on behalf of the respective breed societies according to those rules and regulations. And to do this, we have arguably the world's leading software to support us. And the software is called ILR2 International Livestock Registry version 2. And it's an animal, it's an animal registry service providing for data recording, maintenance, inquiry, and reporting. It is developed by ABRI, Agricultural Business Research Institute in Australia, which is closely allied to the University of Armidale, where one of the top animal breeding houses in the world is. And then, of course, so you've got animals coming in, birth notifications, transfers, but also we now have a process of cancellations for a variety of reasons. I've downgraded it to commercial, it died, I sold it to a commercial breeder, okay, and these we can also cause disposal or fates, and very simply a fate code, one is died, the date, and you send it to ZHB, we put that into the database, and the status of the animal is upgraded. The animal remains on the database, but it's no longer active. And the other way an animal can enter the herd book is through an Appendix A, depending on the rules and regulations of the separate herd books. Okay? And an Appendix A animal is an animal that is born to at least one commercial or unknown parent. It's first birth notified as commercial, and after inspection, it can enter the herd book as an Appendix A if your breed society has an upgrading system. When your birth is notified, the first thing you do is you get a national registration number, and that remains with you for life. This is the same thing here, the system of animal identification. It must be a unique number in Zimbabwe and is used to identify that animal and all its movements and all its activities. It is also known as the Society ID, and the format is very simple. It's 2 digits for the year of birth, 4 digits, 0 padded for the number, tag number within year of birth, and then up to 3 letters being your herd designation letters. And we give an example there, 210036 ABC. So typically, this would mean an animal born in 2021, it's number 36 in 2021, so you need a unique tag number within year, and it's 0 padded for the computer. Okay, it's always 4 digits, 0 padded, and And then we have up to 3 letters being the herd designation letters. So the herd designation letters are very, very important. When you register your stud, there are 2 key fields. The prefix, being the stud name, and your HDL, herd designation letters. So the prefix identifies your stud. Reed Brahmins, holistic Shangani and Cornies, so on. Okay? The HDL is 3 letters that are unique, up to 3 letters that are unique in Zimbabwe, that are associated with each ID. Okay, so anything with ABC is in that stud. So when you register, you need to think about the HDL, because not only is it part of the ID on the computer, but you can also legally put the HDL brand on the animal. Okay? And typically you'll see on the left leg, a hind leg, so Reed Brahmins is REE, and you'll often find Mr. Reed identifies all his Brahmins with an REE, and then he will have 2 digits for the year and 2 digits, the tag number. He won't put, and he will brand that on the animal. You can do that. Okay? Ladies and gentlemen, this is in the constitution of the ZHP, and it is in your society constitution. Births must be registered within 60 days. Why? Because there are so many events tied to that birth, and if you delay on that, it messes everything up, and it messes up the integrity of the data. Okay? Because as I mentioned, the minute an animal is born, what happens? It's birth notified. We link that to that cow. This cow has now given birth. This sire has now given birth. And there's a whole bunch of checks. Was the cow alive at the time of birth? Was it present in your herd at the time of mating? Was the sire alive at the time of mating? Was the sire in your herd at the time of mating? Okay, all these checks. And that's why we want this within 60 days, so that we can carry out these checks, tidy up, giving me a birth at the time when you're selling an animal after inspection. We've tripped up not only Brahmins, but many places where animals are being sold as a stud, pedigree, or registered animal, but it doesn't exist in ZHB, or its status is not clear. So what we are demanding, please, from now on, before you advertise any animal for sale that you're saying is stud, registered, or pedigree, because the minute you use those words, it means it's registered with ZHB. So before you do any sale like that, please get a clearance letter from ZHB. It's very simple. You write to, send an email to Trace. So Trace, Trace, at lit.co.zw, goes to all the data staff in the office. Okay? Please, to the nearest kg. Now, we can submit 2 weights for each trait, and the computer will allow that and take it into account, which allows for any abnormal variances in the 2 weights. So if we, for example, carve from if we've got a typical December to March mating, and we're carving late September to early January, and we wean in May, record the first W2 in April, record the second one in May on or at the same, just before weaning, you've got 2 weaning weights. Okay? We can do the same for W4 and W6. So you can do April, May this year for the weaners, and next year you do the April and May again for W4, and then you can do September and October for W6, for example, if you've got a fixed mating season. However, the system will take multiple weights for mature weight at weaning and cow weight at calving. And again, we are very flexible how we take that data. We also have that very simple Excel, cow ID, calf ID, trait, W2, date, and weight. Very simple. You've told us, so you've mated a whole bunch of cows. You've told us the bull in, bull out. Its calving date, and those 10 cows that failed to calve, you tell me they failed to calve. Because that's what the system needs to know to close. Out of those 30 animals that were mated, 20 calved, 10 didn't calve. That information is sent to us, and that information is analyzed, and we'll get EBVs for days to calving, which is our best estimate of fertility along with scrotal circumference. There are other traits that are being looked at that we need to consider, and some societies are pushing them. Hip height, very good indicator of mature size, and again, if you want to monitor if your cows are getting too big or too small, that's very good. Flight score or temperament. Okay, we can analyze that. It's a very simple record to do it, and some breed societies are pushing it. The Brahmins already have a very good protocol, and it's quite simple. You sit at the crush, and when you let the animal out, you monitor it, how it's going from A to B, and you record it on a score of 1 to 9, if it's nice and calm, or if it's very agitated, and you record that. There is a genetic predisposition to temperament, and if temperament is an issue, you can include it, and you can select for it or against it.
So what we're just going to discuss now is basically why we take that sample and what it's used for. So what we currently use it for is parent verification. So we're talking about multi-size. So you've got a group of 3 bulls, you don't know who the actual parent is. And this is one way of confirming that, yes, such and such a bull is the proper parent of this calf that was just born. Another way is just also to ensure that it's a proper stud animal. It's not from an outside bull that, for instance, jumped the fence or something like that. And they can properly confirm to any possible buyer or ensure that when you're using that bull again or its progeny, that you're continuing the correct pedigree stud lines for its progeny. So what we currently use are microsatellite markers. So I'm not going to discuss that right now, but it's essentially a type of something within the DNA that we use that's passed on from a parent to a calf. And because those have they're shared between the parents and their progeny, we can tell, yes, that this calf belongs to this parent. Okay, and for that we need to take a sample from both the calf and from the parents. So the parents need, say you've already done this microsatellite parent verification for the calf when it was younger, for its parents. That calf will now have a profile at that lab that can be used for when it has progeny, essentially. Okay. So yeah, once the DNA sample's been processed, so whether or not it was a calf or a parent, once it's been processed, it's got a profile on that lab and can be used for future progeny parent verification for future progeny. Okay. All right, we take currently 3 different types of samples, okay, to store at the ZHP offices. The most common one is the pooled hairs, 20 to 30 with the follicle. So with that bit at the end, that's where the actual DNA is, not in the hair itself. So you want to make sure you pull it and you get that tissue that comes with the hair. Because if you just cut it, there's no DNA in there. So make sure you take 20 to 30 so you have a good sample size for processing and can be processed several times if needed. What we currently have are envelopes with animal details written on, and those can be sent to the lab. What we're kind of moving to, as some of you might know, are these specialized hair cards. They're basically these hair cards. You take the hair, you put them in, you flatten it so that the hair is secure, it can't be opened by anyone else. In order to process it, they actually have to take punches of this card. So there's no handling or any cross-contamination with any other types of DNA, whether it's human or other types of hairs. Another type of DNA sample is a tissue sample. So there's specialized tags where when you put the tag in, it takes a section of tissue and places it in a vial with a preservative in it. And because the vial is fairly small, the animal ID is written on it and can be linked up with the actual animal details itself. Okay. Currently, if anyone's with nurture, finance, this is how they currently take their DNA samples in conjunction with the electronic ID. And because you've got those two things, you can always identify the animal should something go wrong. So you've got the DNA sample in case the animal loses all its IDs, identification, ear tags fall out, brands wear off, or overwritten. Once you've got a DNA sample stored at ZHB, we can take that sample itself, and you can take a sample from the animal. You can confirm the ID as well, that the animal is what you say it is, even if all possible visual identification is lost. Okay, levulus. Okay, so each year, because ZHB curates the data of all the stud breeders and maintains their records, records their performance data, does analysis, all that. That needs to be funded, and that's done through levies. Okay, and basically that's done according to the amount of animals you have, as well as standard levy, herd levy on top of that. So you have your herd levy, and then plus however many registered or calf book animals you have that were born before the previous year. For instance, 2022 levy list will include all animals born before the 1st of January 2021. And that only applies to your calf book and registered animals, active animals. So you can have as many commercials or commercial animals as you want. Those aren't levied because they won't be producing any calves, and ultimately they're just there at your own discretion. Okay. We can generate a levy list in PDF format or in Excel format. Most people find that the Excel format makes it easier to go through it and add your own notes to it. However, the PDF format is quite readable. Especially if you print it out, you can easily make marks on it. However, if you have been keeping up with your birth notifications, there really shouldn't be any missing animals on your levy list. Okay, so members need to confirm that the animals that should be there are there, and that the details match with what you have. So you need to make sure that a cow's progeny number, however many times she's calved, that's accurate, her ICPs are accurate, her age of first calving is accurate. You need to confirm that all the details are there so that you can catch something before it starts snowballing and ends up messing up all your data. Because you're suddenly looking for 4-year-old calves. So now genomics, exciting stuff. Lots of talk about it in the past few years. But what exactly is it, and how is it going to help us? Okay, so what we currently do in terms of genomics, we have how we currently analyze it. What we're currently working with are microsatellite markers. As we discussed earlier, it's how we do parent verification currently. What actually are they? Okay, so what they are, they're pieces of DNA with repeats and high mutation rates found throughout an animal's DNA. So that basically means they're these specific types of repeats that are found multiple places throughout an animal's DNA. But one thing to remember is that these repeats and the places where they're found in an animal's DNA are passed along to their progeny, essentially. So if you know where these markers are in a sire, they'll most likely be in the same place for its progeny. And that's how we do parent verification. Okay? What we're currently using are these 12 standard ISAG markers, so International Society for Animal Genetics, and they're used for parent verification. So they're, like I said, specific places, specific repeats. If the repeats match what's in the parents, they'll be found in the progeny. So just remember that's how it works. Okay? Because there's various steps to doing it within the analysis, and each lab can have its own unique steps and calibrations and all unique set of equipment and all that. It's not very easy to repeat the results between labs. So it's not always possible to get an animal's DNA processed at one lab and then take the results or the profile to another lab without them first sending a whole bunch of details as to how they processed the DNA, what additional steps they took, or what. Basically, it's very difficult to standardize these results between labs, but it's possible. But ultimately, if you process the DNA of an animal at one lab and you want to do its progeny, best bet is to send the progeny to the same lab for an easier and faster results. And also the actual analysis, because it requires a whole bunch of first go get the DNA out and then process it in a certain way. It's very time consuming, and to basically extract the DNA, to cut it up, and then to analyze it for those markers. Which is why we have now, people are pushing these single nucleotide polymorphisms, or SNPs markers. So there's lots of them spread throughout an animal's DNA at specific positions, okay, and different variants of these SNPs exist, and they can affect an animal's performance. So these SNPs are found in specific genes, and these genes are what affect an animal's performance. So one can see that if it has a certain SNP within that gene, it will affect an animal's performance in such a way. And that's kind of what we're working towards with getting this whole big genetic profile for breeds and for entire breeds, essentially, is to find these different variants and how exactly they relate to an animal's performance, so that if we find these variants in a new animal, we can make a good estimate as to how their performance will be affected. Okay? Compared to microsatellite markers, they're relatively easy to measure because it's a whole different process to actually analyze them, and they can also be used for parent verification, because if a parent has these SNPs, their progeny will most likely have the same SNPs as well. But other than parent verification, they can be used in other applications. I'm going to go through here. So one application is generating these genomic estimated breeding values. So what that means, as I discussed, if a trait is linked to a certain SNP, if an animal in the future has that SNP, we'll know that its performance will be similar. Also, because those SNPs are basically, you can use them to start identifying not just the parents, but we can start looking at siblings, uncles, aunts, grandsires, all that. Because these SNPs are more widely spread throughout an animal's genome, there's more of them. We can find more accurate pedigrees, meaning that we're no longer going to have gaps in the pedigrees. We can start linking individual animals to each other and how exactly they're related. Okay, so also the benefits of these genomic EBVs. Okay, so you can take a DNA sample from a young animal before it's produced any performance data. I mean, in some cases, even before it's born, and you can get EBVs for that based on what its DNA is telling us.